So good morning, everybody, and welcome to Gem Pharmatech's monthly webinar series. My name is John Jackson. I'm part of the marketing team here, and I'll be your host today as Dr. June Lee, head of our preclinical oncology pipeline, does most of the talking, if not all the talking, on this topic of innovative mouse models to accelerate the development of in vivo CAR T therapies. Before we do that, just to give you a quick rundown if you are unfamiliar with Gem Pharmatech, we are a preclinical CRO, and our mission, very simply, is to accelerate research and discovery for scientists across the globe. The way we do this is by offering unrivaled access to in vivo products and services. This includes over 25,000 genetically engineered mouse models, the knockout portion of which was created with the specific purpose to give as many scientists and researchers as possible access to models with their target gene of interest. We have a huge capacity to generate new models every year. And obviously, this webinar is focused on oncology. And Dr. Junli will talk about some of our humanized and immunodeficient strains. But I encourage you to reach out, to follow us on LinkedIn, to continue attending these webinars, to learn a little bit more about the other stuff we do, and do not hesitate to reach out if you have interest in a different area. With that, I will leave it to Dr. June Lee to begin the scientific portion of this webinar, and hope that everyone stays until the end to participate in the Q&A session.
Good morning and good afternoon, everyone. So I'm Jun Lee. I'm the head of the preclinical oncology at Gem Pharmatech. So today, I would like to talk about how the innovative mouse models are serving the critical bridge to the next generation of the in vivo CAR T immunotherapy. So my talk would be include 4 parts: the in vivo CAR T as a emerging trend, and the classical immunodeficient xenograft models, and the selection of the mouse models for in vivo CAR T, and the challenges and the future directions. So to begin with, to understand the preclinical demands, we need to look at the clinical landscape. So we know that the FDA has approved several the CAR T therapies targeting the CD19 and the BCMA. Most of the indications are lymphomas or multiple myelomas. So, however, these are all ex vivo therapies. No single in vivo CAR T has been approved by the FDA so far. So the ex vivo CAR T therapies face massive hurdles. For example, the total procedure cost for one patient can up to for $1 million. So it's very expensive, and not all the patients can afford this therapy. So compared to the ex vivo therapy, which has very high efficacy in the hematological malignancies, but the high cost and the complex manufacturing, and also need the patients to delete the lymphocytes, can be quite long time costing, and also the high cost in terms of the expenses. For compared to the in vivo CAR engineering, which means the CAR is reprogrammed, the immune cells is reprogrammed inside the patient's bodies using vectors like the viruses or LNP. So the advantage compared to the ex vivo CAR T, it improves the accessibility and shorten the time. And also, more importantly, it reduces the cost and the logistics. Also have the potential to lower the toxicity as well. It overcomes the manufacturing bottlenecks with a higher efficacy and persistence. So in the clinic, if you want to design in vivo CAR engineering, there's a number of key factors you need to consider. For example, let me get the laser. So for example, first you need to choose what target you need to, what target you need to design. So the target specificity is very important. Another point, key factors, you need to choose the delivery system. Which delivery system you need to use must align the therapeutic context and also what the payload you need to use. The other key factors, such as the transgene design, which will influence the safety and the efficacy as well. So require the specific immune cells promoter and optimize the CAR architecture. Also, we need to consider to minimize the immunogenicity by either overexpress, for example, the CD47, or to knock out the MHC class 1 or class 2 to evade the immune detections. And safety is also important to monitor, such as the CRS or the ICANS for potential and the potential insertional mutagenesis as well. So for the preclinical stage, we need to use the mice models to assess the efficacy of the in vivo CAR. So what kind of mice models we need to choose to have a successful in vivo CAR engineering in the patients? So first, we need to, because the immune system is human, so we need to choose as a basis, the foundation of the mouse model is the immunodeficient mice. For example, the NCG mice, which is knock out the PRKDC and the IL-2RG based on the node stream. This NCG mice is a triple immunodeficient strain. It lacks the T cells, B cells, and NK cells, also with reduced macrophages as well. So it allows for a high engraftment rate of the human cells. So, and it makes the NCG strain ideal for development the humanized mouse models, either via the CD34 human stem cell engraftment or the human PBMC transplantation. So this humanized model has a wide application areas. For example, it can be used for oncology study to do PDX or CDX, also like immunology or infectious disease can be used in this strain as well. So the NCG is the foundation of the immunodeficient mice. And based on that, we have also developed the refined NCG variants to solve specific experimental problems, such as the NCG-M strain. And we genetically modified this strain to express human cytokines, like human IL-3, human GM-CSF, and the CSF to support a better myeloid lineage engraftment. Also, we have developed the NCG MHC-1 and 2 double knockout mice, which significantly delay the GVHD in the PBMC reconstituted models. So the NCG, if we look at the picture on the right, it has no T, B, or NK cells with only reduced myeloid cells. So it's the most immunodeficient strain compared to NOG and NSG. So why the mice need to be humanized? Because the reconstitution of the human immune system would better mimic the human cell T cell responses and also the cytokine release syndrome and off-tumor effects as well. So basically, there's two types of humanized mice models. One is the human PBMC reconstituted NCG. This model has a rapid reconstitution rate and a short time to reconstitute. It's very good for acute responses to assess the T cell functions, but it has a GVHD risk. For the human stem cell reconstituted NCG, so the stem cells are CD34 positive. It can build longer time reconstitutions, and with multi-lineage of the reconstitution. For example, it has T cells, B cells, and myeloid cells as well. So the in vivo CAR, excuse me, application can be used to test either the viruses vectors or LNP vectors delivery to the human T cells for the reconstitution. So the advantage of using human reconstituted mouse models, it can recapitulate the human CAR T kinetics and persistence, and also can be used to evaluate the adverse effects as well. So here we're just showing the levels of the immune cells reconstituted in these two different types. So on the left is the PBMC reconstituted NCG mice. The major cell type reconstituted is the CD3 positive cells, and also the CD8 and the CD4 positive cells. The CD3 positive cells can be reconstituted up to more than 90% in the peripheral blood. On the right side is the HSC reconstituted mice. And it's not only have like more than 20% of CD3 positive cells, it has also reconstituted the CD19 positive cells, which is B cells and the NK cells as well. For the NCG, for the HSC reconstituted NCG mice, it has very long survival time, can be up to 200 days of the reconstitution compared to the GVHD happened in the PBMC-NCG mice. It has very long survival time. So another part for the in vivo CAR T is the delivery system. Basically, there's like 4 types of delivery system. One is the LNP formulated nucleic acids. It contains the either mRNA or plasmid DNA. The other one is the polymeric nanoparticles that encapsulate the nucleic acids. And also the viral system, either the lentiviral system or the AAV-based viral delivery system with the target on the virus surface, such as the scFv targeted specific receptors. Just to give you an example, this experiment have done in our facility. We use the NCG MHC double knockout mice inoculated with the NAM6 human leukemia cells and gave the PBMC to reconstitute the immune system. Then we gave a single dose of the lentivirus targeting CD3 cell subsets in this PBMC reconstituted double knockout mice. And then we monitor the peripheral blood CD3 percentage as well until day 80. So this is the results of the tumor growth. You can see the treated group, the green line has a significant tumor growth inhibition compared to the control group. Also, the survival rate is much longer than the control group as well. Here, there's the live images of different days of the mice. In the treatment group, you can see there's basically no tumor growth in the treatment group. So the human PBMC-NCG double knockout MHC mice significantly reduced the GVHD occurrence and minimize the xenogenetic responses caused by the active T cells. Another example using lentivirus is the VivoVac system designed by the Umoja Biopharma. So the lentivirus is the characteristics of this is it has very high transduction efficacy, and it integrates with the genome to ensure a stable CAR expression. So the LV must be pseudotyped to avoid the target sequestration in the liver and ensure the specific T cell targeting. So this platform designed by Umoja Pharma utilizes a surface engineered lentiviral particles with anti-CD3 scFv for T cell activation and targeting. So the A figure shows the study design. and they use a NSG MHC-1 and 2 double knockout mice inoculated with NALM-6 tumor with luciferase tag at day -4. And on the day -1, they gave 20 million PBMC reconstitution by IP injection. On the day 0, they dosed the mice with UBV 100 by IP injection as well. Then they monitor the tumor growth and also take the blood samples to look at the T cell activation and T cell count in the peripheral blood. So the B figure shows the results of the T cell activation at day 4 by injecting a different amount of the lentivirus. It definitely shows a dose-dependent activation of the T cells. Also, it shows in per microliter of the blood, the T cells increases and reaches the maximum at day 11 and gradually decline afterwards. By looking at the efficacy, you can see the tumor burden the tumor growth has been significantly inhibited in the high doses, the two lines at the bottom, the orange line, the red line. Also, the high doses treated group has the longest animal survival. Another viral delivery system is the adeno-associated virus, or the AAV-mediated gene therapy. So the AAV is different from LV because it's non-integrating, so it leads to the episomal expression. It minimizes the insertional mutagenesis, but the pitfalls is it results in the dilution of the CAR gene when the T cells undergo proliferation or division. The AAV9 serotype exhibits a broader tropism and a higher efficacy in the hematopoietic tissues. When it combines with the IL-15 cytokine, it enhances the overall reconstitution of the CD8 T cells function in the NCG mice. Also, I'll show the results in next slides. The AAV encoding the anti-CD4 CARs successfully reprogrammed in the human PBMC reconstituted NCG mouse model and leading to tumor growth regression. So here the results. So the study design is like this. So they gave the PBMC to the mice at week -3, and then they inoculate the tumor cells MT2 at day 0, and gave the AAV CD4 CAR at day 2, and do the analysis from day 3 to day 4. So if you look at the figure B, compared to the untreated group at different days, the treated group with CD4 CAR significantly reduced the tumor burden in the mice. And it shows here the quantified results. Compared to the untreated group, the tumor growth are increasingly dramatically, but the treated group has about 3 out of 6 mice showed complete tumor regression. When they monitor the CD4 positive cells in the blood, so they compared the only PBMC reconstituted mice without tumor. This group is tumor bearing mice, but without treatment, without the CAR CD4 CAR treatment. So you can see in the treatment group, basically there's no CD4 positive T cells in the blood. It's 0%. Because and in the untreated group, it has 78% of CD4 positive T cells. But in this no tumor group, it has 56%. When you look at the CD8 positive T cells, the treatment group definitely increased the CD8 positive T cells in the mice compared to the untreated group. Also, the depletion of the CD4 positive CAR in the peripheral blood compared to the untreated group. They also look at the biodistribution of the CAR in different organs. They look at the lung, the heart, liver, kidney, spleen, intestines, and the reproductive system. Basically, the AAV-treated CAR only accumulated in the intestines. So the most popular delivery system is the LNP mRNA system. So the LNP stands for the lipid nanoparticles, offer advantages in safety and scalability and the ability to repeat the dose. The tLNP platform designed by Capstan Therapeutics now is part of the AbbVie. They use the LNP to target CD5 or CD8 for the delivery of anti-CD19 CAR mRNA. Also, these ionizable lipids, such as the L829, designed with the tLNP platform together, minimize the delivery to the liver. Because we know that if you're not the targeted LNP, just the LNP, it's always accumulated in the liver. And this design would maximize the uptake in the spleen and the lymph nodes for the immune cells and reduces or minimize the uptake by the liver. And the NCTX platform uses the targeted LNP to deliver mini-circle DNA and the transposase mRNA for a stable genomic integration. So the results shows that a single dose of the NCTX induced a stable CAR T, about 100 cells per microliter of the blood in the preclinical stage by using the human HSC reconstituted mice. As we know that this in vivo CAR designed by Capstan, this CPTX-2309 are now in first stage of clinical trial. Now here I give you an example of the tLNP encapsulating the B cell targeted CAR induces the B cell depletion in the HSC reconstituted NCG. So here's the structure of the ionizable lipids L829 tLNP targeting CD8. If we look at the flow cytometry data of the CD8 T cell expression, they used in the spleen, they used the human PBMC reconstituted NCG mice after the administration of 30 microgram of tLNP. So the CD8 T cells expression CAR increased until day 6, and sorry, this is 6 hours and not day 6, and gradually declined by 24 hours. Because the PBMC reconstituted NCG do not have the B cells, so in order to look at the B cell depletion, they use the HSC-NCG mice to look at the B cell depletion. So compared to the control group, which is the yellow lines, the blue lines showed the dramatic B cell depletion in terms of the percentage of CD45 positive cells. But gradually, after 14 days, the B cells start to regrow again. In the efficacy study, they used the human PBMC reconstituted mice again, inoculated the mice with NAM6 tumor, and took the images at day 2, 6, 9, 13, 16, 21, 23. And the dosing of the LNP, because they need the repeated dosing, so they dose them at day 0, 3, 7, 10, and 14. So the image shows here by the live cell bioluminescence imaging system showed in this PBMC-NCG mice, the treated group, they treated 2 different doses. One is 10 microgram, one is 30 microgram. So the 30 microgram basically shows no tumor burden at all until day 23. Another example for the targeted LNP mRNA system using the human stem cell reconstituted NCG mice is this NCTX model. So this is the structure of NCTX, and they use the SB100 transpontase mRNA, and targeting encoding the mini circle DNA inside of it and targeting CD7 positive T cells. So the study design is use NCG reconstituted with human CD34 positive cells, also inoculated the NAM6 leukemia cells, express human CD19. Then afterwards they gave IL-7 to support the T cell proliferation, and they gave the treatment of the in vivo CAR. And then they monitor the tumor growth twice a week. So the results shows here in the B. Sorry, this is the CAR T cells. How many CAR T cells per microliter blood? So this CAR, sorry, I should mention, this CAR is a dual CAR, mini circle DNA expression. They express CD19 and CD22 as well by flow cytometry, up to 40 days. So you can see that the treatment group have dramatically increased the CAR in the blood compared to the control. If you look at the image of the live animal imaging in the treatment group, the tumor burden, they have 12 mice in the treatment group. They have 6 mice in the control group. So the control group basically died or euthanized after day 21, but the treatment group can still survive until day 42. Here is just the survival curve compared to the two groups. So definitely the treatment group with the CAR can survive up to 42 days. Another example of the targeted LNP, they just use the syngeneic mouse models. So they injected the CAR into the wild type C57 black 6 mice and monitored the CD3 and CD7 in the blood. So this LNP targeted CD3 and CD7, carrying this CD19 CAR encoding mRNA. So they gave 2 different doses of each. So CD3, they gave low and high dose, CD7 low and high dose, and the B10 is just the untargeted control group. You can see by 12 hours, the CD3 dosed group have the highest CAR in the blood, and also have the highest CAR of the fluorescence intensity, the MFI as well. When you look at the B cell depletion, you can see the B cell depletion by the CD19 specific CAR T activity as shown, the decreased B cell percentage in the single cell population. And so the highest bit is after 36 hours compared to the PBS group. Another model you can use is just use the NCG mice, such as the CDX model. So you inoculate the HCC-1954 to the NCG mice, and this target, the MT302, is encoding CHOP2, which deliver to the monocytes. So you inoculate the immunodeficient mice at day 0, and you let the tumor to grow and until just day 22, and you give the treatment every 4 days, so for 5 times, and the tumor growth is slightly inhibited at the dosage of 1 milligram per kilo. And when you look at the CAR positive monocytes in different organs, such as in the blood, in the spleen, and in the bone marrow, it all dramatically increased compared to the control group. And also in the tumor, you can see there's accumulation of CAR positive monocytes in the tumor as well. And then they changed the dosing frequency by a biweekly injection of this in vivo CAR. So they gave them 3 times biweekly, and also shows quite significant tumor growth inhibition as well, as shown at the red line. So just to summarize that which mouse model to select and what's the difference between these models. So the NCG is the basis foundation of the immunodeficient mice. Without any reconstitution, it can last. So the study can last for like a year long, and there's no risk of GVHD, etc. It's usually used for CDX models and the PDX models. So for the PBMC compared to the HSC, it has mature T cells. The HSC has multi-lineage. It has TB and myeloid and NK cells. But because the high risk of GVHD, so the study timeline is relatively shorter and up to 3 to 6 weeks. But the HSC has up to 6 months and with very low GVHD risk. So if your target is human T cells, you probably can choose the PBMC reconstituted. But if your target is NK or myeloid cells, you can consider HSC reconstituted for longer time. So this is fast model to test the T cell directed LNP, and this is more like clinical relevant. You can test like on target off tumor safety as well. So the limit for the PBMC model is GVHD limits the observation time. For the HSC, it's relatively expensive. It takes longer time to prepare, and the myeloid cells is relatively low as well. This is the comparison of different delivery cargos. So the lentivirus has a stable integration, and same as the retrovirus. The AAV doesn't have the integration as well. Doesn't have the integrating to the genome. The LNP is transient. The expression is very transient in the LNP. It's transient in the AAV dilutes with the division, but these two are permanent. So the targeting cells, the LV can target dividing or non-dividing. The RV target dividing only, and the LNP can target any cells. And this is the size of the cargo capacity, and also have the safety risk. So, so the LV has the insertional mutagenesis risk, and but the LNP, the popular one, has quite high biocompatibility. So, and for the safety and toxicity evaluations, I think the CAR mediated cytokine release toxicity is related to the surge of the T cell activation. Then they release the cytokines such as IL-6 or IL-10, which in turns to stimulate the myeloid cells to further release the cytokines like pro-inflammatory cytokines as well. So in the xenograft model, because the interspecies cytokine cross-reactivity is not complete, so the host immune system is inadequate. Basically, so the cytokines is sometimes is mouse, but you use the human and T cells, so they do not interact with each other. But for the CRS and the HLH, the side effects, there are reports that say they successfully modeled in the syngeneic and the transgenic immunocompetent mouse models. For the neurotoxicity and the CRS modeling, I think it's more relevant to human is to use non-human primates to mimic the human toxicity, and it will provide more valuable insights. So to conclude that the targeted ILV system, so it demonstrates very powerful stable integration for long-term B-cell aplasia and tumor control. The most popular, the LNP and the target LNP mRNA platform provide a safer and redosable alternative, and it not only works well in the oncology area, but also expanding rapidly to the autoimmune and the infectious disease as well. So the NCG, this variant strains refines our ability to predict the toxicity and optimize the therapeutic window. So for sure, the humanized NCG platform remains the primary benchmark for assessing the off-shelf immunotherapies. So for the future directions and the model improvement, I think we can think about maybe to use the pluripotent stem cells, derived cells. It may have better cytokine transgene for the innate cells as well, because we would like to, as close as possible, to reconstitute the immune system close to the human. And we also can combine the syngeneic models, like you should use the mouse CAR constructs in the immunocompetent host to study the tumor microenvironment and the T cell exhaustion, for example. And we can also use the multi-omics to track the T cell dynamics and the biodistribution. And also, we know that non-human primates for safety and the PK analysis is more close to the human. So, but the goal is always the same: to build models can predict the clinical success of the in vivo CAR in cancer and in autoimmunity. So, last but not least, so Gemma Pharmatech supports global researches by, for example, we have published 2,731 papers in the peer-reviewed journals, and the total impact factor more than 26,000 in different research areas from immunology, oncology, metabolic disease, etc. And this is just an example what journals we have published by supporting the research of globally. We're not only just providing the mice models, we're also a CRO company to provide the preclinical pharmacology evaluations as well. And thank you very much for attending this seminar. If you have any questions, you can submit your question to our scientific team, and we would like to answer your questions. Thank you.
Thank you, Dr. Jun Lee. We'll certainly try to answer some of your questions now, and I'll begin looking over those. I'll give everyone maybe 20 or 30 seconds to type one of their questions.
Okay.
I have a question here. When evaluating a new in vivo delivery vector, how do I choose between the humanized PBMC-NCG and the humanized HSC-NCG models?
So the difference between so it depends on your study goal and the duration and the complexity. So the human PBMC is best for like a rapid efficacy screening. It provides like high frequency of the mature CD3 positive T cells, so make it ideal for like short term, like a week to 4 weeks studies for the T cell directed LNP or viral vectors. So the HSC-NCG is better for longer term persistence and safety. So because it has a relatively full multi-lineage reconstitution of the human immune system, it has TBNK and myeloid cells. So if you need to observe the CAR T persistence for more than like 40 days, for example, or evaluate the on-target of tumor toxicity, probably the HSC-NCG is necessary or a better choice.
Thank you. Just so everyone knows, we will be taking down these questions in case we don't get to them right now. Let's switch it up a little bit. Okay, one about graft versus host disease. So graft versus host disease often limits, I think they mean study window in xenograft models. How does Gem Pharmatech address this for these long-term in vivo CAR T studies?
Yes, so if you use the PBMC reconstituted NCG, you can't avoid the GVHD because it happens quite quickly after probably 4 weeks. but we have a solution by developing NCG, MHC class 1 and class 2 double knockout strain. So in this strain, by removing the MHC 1 and 2, and it significantly delay the recognition of the mouse tissues by human T cells. So it definitely extends the therapeutic window and compared to the standard NCG mouse. So it allows for longer term tracking of the CAR T expansion and the tumor monitoring without worrying about the GVHD. So
I think we'll do one more. I have a very, I don't know, seems like a general question. When should a researcher use a syngeneic model, like a basic Black 6, instead of a humanized NCG model for in vivo CAR T testing?
So if your target, if you would like to, because the syngeneic model, the difference between that with the reconstituted model is the immune system is intact, it's immunocompetent, it has all the natural lineages, development of the immune cells. But your CAR, I think the design need to be targeting the mouse CAR instead of the human CAR. And it's ideal for monitoring either like toxicity or long-term CAR T persistence. But most of the biotech or biopharma, they design the CAR specifically to target human immune cells rather than mouse. So I think you most of the time you have to choose the reconstituted immune system strains instead of the syngeneic models.
Okay, just a reminder to everybody, if you did ask a question that wasn't answered on here, which there are quite a few in the chat, we will send you an email with an answer to that question. And alternatively, you can reach out to sales@gempharmatech.com. I thank everybody for joining. Next month, we will be giving a webinar on our knockout model collection, as well as custom model generation. And we look forward to seeing you there. Everyone have a wonderful day and follow us on social media. And again, reach out if you have any questions at sales@gempharmatech.com. Thank you.
Thank you very much.